everyone, welcome to the channel Bioverse with Pallavee. In the part 1 of this chapter, previously we had discussed about the floral primordium, floriculture, structure of flower, and the male reproductive unit that was stamen. In today's class for chapter sexual reproduction in flowering plants of class 12, we'll discuss about the stamen microsporangium, microsporogenesis, and various topics that are related to pollens. So let's get started. So the very first thing that we are going to study is the microsporangium. And we have on that day discussed that the microsporangium refers to the pollen sac. So total of 4 pollen sacs are going to be present. In this diagram, children, you see that this layer is the outermost layer, an outermost protective layer that we call it as the epidermis. So we need to remember that this layer is going to be only single celled and it is protective in nature. Then the next layer that has been marked over here, children, this is called as the endothecium. Endo means towards the inside of the epidermis. Again, this layer is also going to be single celled, and we need to understand that this is the layer that is going to be specialized for dehiscence. Now, a very new word we are listening is dehiscence. What do you understand by dehiscence? It refers to the spontaneous opening of the pollen sacs, that is the microsporangium, at maturity, and it releases the pollen grains. So, when we talk about endothecium, this is not only radially consisting of the single cells, but it also consists of cells that are radially arranged and are elongated. Now, when these radially elongated cells which form up the endothecium they are present, they also give us the protective strength, a mechanical strength that helps in the dehiscence. Now, how we are going to get this mechanical strength? It means that some kind of chemical is going to be present. So, for the chemical that we are talking about is alpha cellulosic fibers. These alpha cellulosic fibers are the actual one that makes it rough, tough, and provides the mechanical strength. Next important thing is until and unless the maturation of the anther takes place, till that time this layer is going to be hygroscopic. Hygroscopic means it is going to absorb the moisture. When anything absorbs moisture, then it supports the growth, supports the maturation, and once the maturation will be attained by the anther, then it will be getting dehydrated and it will promote the dehiscence. Next thing is the middle layer. What needs to be understood is that this middle layer is going to be multi-layered. Now, when it is multi-layered, it is going to be present just below the endothecium. It provides protection and provides support to the microsporangium. Now, when all the growth are been taking place, what is required here is the food for the growth. So the cells of the middle layer, they do the food storage. Now the food storage is being done by the parenchyma cells that are present in the middle layer. So the most important thing to remember is M for middle and M for multi-layered. So the middle layer is going to be multi-layered, it is going to be thick. It is consisting of parenchyma cells that will be helping in the storage of the food. And in turn, it provides protection and support. Now, one more term that I'm going to use over here is ephemeral. The word ephemeral refers to that this layer is going to be short-lived. Now, why are the words short-lived being used for this layer? It is because when the epidermis, endothecium and middle layer of the anther they reach the maturity, they need to be dehisced. They need to be bursted out. There needs to be a crack so that the pollen sacs release the pollen. So that is why it has a very short duration, and that is why we use the word ephemeral. Now, the next innermost layer that we are talking about here is the tapetum. Tapetum is the layer that is going to provide the nourishment to the developing pollen grains. So, whenever we are talking about anything that provides a lot of nourishment, it means that it is going to have dense cytoplasm. Without the dense cytoplasm, nutrition cannot be provided. Yes. So after being having the lot of amount of the nutrition present within the cytoplasm, this layer is also going to be multinucleate. Means we understand that when the tapetum layer was being initially formed, it was diploid, but later on the ploidy has been taken into polyploidy. Polyploidy means here lot of nucleuses are going to be present. So means we need to understand that karyokinesis is happening. But ये जो karyokinesis हो रही है, this is not followed by cytokinesis. तो इसका मतलब कि हमारे पास सेल वॉल की फॉर्मेशन नहीं हो रही है और इस वजह से हमारे पास बहुत सारे न्यूक्लियस बन के तैयार हो जाएंगे. Next important thing that we'll be discussing in another slide, but the term reference that we're talking about here is sporopollenin. Sporopollen is a structure that is going to be secreted by the tapetum layer. Now, one more important thing is pollen kit. This pollen kit is a structure that is present as an oily layer around the pollen grain. The plant in which pollination generally takes place by the insects, there these pollen kits are present and they are composed of lipids and carotenoids. So all in all, the function of the pollen kit is the oily layer protects the pollen grains from the harmful ultraviolet rays, and since they are being having the sticky surface and their oily nature, it helps them to attach to the body of the insect, and since they are yellow in color due to the presence of the carotenoids, it attracts the insect, and because of that the pollen grains have been carried from one place to the another. Now, this purple color tissues that we are talking about is sporogenous tissue. A group of compactly arranged homogeneous cells that are present in the center of the tapetum are called as the sporogenous tissue. These tissues will give rise to the microspores, that is, the pollen grains. So let us understand, children, that just in between the center, we are going to have the connective tissue. This connective tissue consists of the vascular bundle, and together with this, all these four sacs, means all the four microsporangia within the anther of the two lobes, they are connected to the filament through the help of this connective tissue. A typical anther is thus dithecous tetrasporangiate in the transverse section. Each theca, now theca refers to sac. So here we are talking about the microsporangium or the pollen sac that consists of two microsporangia per lobe, and thus in all we have total of 4 microsporangia in the anther. Each microsporangium is then being surrounded for 4 layers of walls that are called as epidermis, endothecium, middle layer and tapetum. Next, we'll understand what is microsporogenesis. The word genesis refers to generate, and when we are using the word microsporo, here we refer to formation of the male gamete. So the formation of the male gamete is called as the microsporogenesis. Now, when we need to understand what is microsporogenesis, what we see is that the innermost layer was the tapetum. Yes, so let us understand this is the tapetal layer. Okay, okay, children. This becomes the tapetum. Now, inside this tapetum, what happens? A lot of sporogenous tissues are going to be present. Now, these sporogenous tissues are going to have what kind of ploidy? Diploid. So these sporogenous tissue are first of all diploid in nature. Now, what happens is these sporogenous tissues, they are going to differentiate themselves, and when the differentiation happens, it is happening because of the division that is called as the mitosis. So till now what we see is mitosis is going to happen, and as a result of which what is going to happen is the tapetum layer with a lot of sporogenous tissue, they are also going to have the differentiation into the microspore mother cell. Since it is forming the microspore or the male gamete, so we use the word microspore mother cell, or even sometimes children we use the word pollen mother cell. So that is why sometimes you have seen the abbreviation as MMC or PMC. We should be in habit of writing the complete full form in your copies when you're giving your answers in the examination. Avoid using any kind of the short forms. Then, when this sporogenous tissue that was present inside the tapetum layer differentiates, it forms the microspore mother cell, but here still the ploidy is going to be 2n. Now, let us enlarge this microspore mother cell. This microspore mother cell with a ploidy of 2n. Now, what we see over here is, now, since we are talking about the microsporogenesis, children, I'll make you understand two terms. First term is gametophytic, and the second word is sporophytic. In the chapter Plant Kingdom, we had studied that sporophytic is the diploid body, and in case of angiosperm, it forms the body of the plant. So sporophytic is the plant body, and gametophytic refers to the body that is going to form the gametes. Gamete refers to the pollen grains in case of the androecium. Yes. So here we are going to see the formation of the gametes, and whenever we are talking about sexual reproduction, we remember that always meiosis division is going to happen, and as a result there would be reduction in the ploidy. So that is why in case of angiosperm what you see is alternation of generation. The alternation of generation is going to happen in such a way that these gametes will fuse to form the zygote that will develop into the embryo, and finally you'll get the full plant body. And this sporophytic body will be diploid, and this sporophytic body, when again undergoes the mitotic division, it is going to form the gametophytic body. So in this way what happens is you will be getting both the male and the female body. Since here we are talking about the microsporogenesis, we are going to see that here the male gamete, that is the pollen grain, it is going to be formed. So here what has happened is the gametophytic body has been formed, means the formation of the gamete is now going to happen. So this microspore mother cell is now finally undergoing a division that is going to show the reduction in the ploidy. The microspore mother cell being the diploid will be converted into what? A structure with a ploidy of haploid, that is N. Now, you'll be having the formation of four tetrads. So what I say over here is, since these are going to form the microspore, let us say that this is the microspore tetrad that is going to be formed out of it. Now what happens, this microspore tetrad, they are not loose. They are being tightly packed with each other with the help of a substance that we call it as the callose. Yes. So for few duration, they are been going to be attached to each other with the help of a substance which is a protein, that is callose. Now, the microspore tetrads can be arranged in various ways, children. Let us see how. If they are arranged like this in this manner, then we call them as linear tetrad. Now, when they are arranged in this manner, just the simple, all the four tetrads, so means they are all in the same plane, so it is called as isobilateral. When they are present in the form of this structure, somewhat T-shaped structure, then it is T-shaped tetrad. But the most common type of the tetrad that is visible is in most of the pollens, it is the tetrahedral one. So this is your tetrahedral tetrad. Now, the fourth one is not visible because here what we are seeing is the 3D, 2D structure. Otherwise, in the 3D structure, it is present towards the backside. Okay, so what we have seen is the tapetal cells, they had the sporogenous tissue that was diploid, and upon the differentiation through the mitosis, they had formed the microspore mother cell or the pollen mother cell. And this was diploid in nature. Now, the microspore mother cell, when it undergoes the meiosis, it forms the tetrad. Now they are attached to each other tightly with the help of a protein called as callose. Now, after some time, what will happen is each of the tetrad of the microspore, they are going to get separated. Now, when anything is getting separated, we see that there is a role of certain kind of enzyme. So, as to break the callose, we have an enzyme that is called as callase. Whenever we see the word ase, it means that definitely it is some kind of an enzyme. So, callase is going to come and it is going to break down the proteinaceous covering of the callose. This callase is being secreted by tapetum. So now all the microspores, total of how many? 4 microspores have been formed. And these 4 microspores, they are going to be what in nature? They are going to be haploid. So these microspores, what do we call them? These are the first cell of the male gametophyte. Okay. So now finally what has happened is we have got the first cell of the male gametophyte. In the next slide, children, we'll talk about how does the pollen grain is formed. Now, the pollen grain that has been formed would be, first of all, a male gametophyte, but that is immature. So this is all about your microsporogenesis. Now, we are going to talk about the pollen grains. Now, in general, what we see is the pollen grains are going to be two-celled. So, for being two-celled, what we see is we have the outermost layer in a form of a geometric fashion. Now, they can be of different types and different kind of the pollen grains. So, children, this outermost layer we call it as exine. This layer that is inner side of the exine, this is called as the intine. Now, Together with that, what we see is children, at certain portions, the exine is free. Here, no such boundary is there. So the structure where on the outer surface of the exine, it is having a very thin layer or it is completely absent, such structure is called as the germ pore. So when we'll talk about the pollen pistil interaction, we'll see that it is only through this germ pore that the intine is going to follow and form the pollen tube during the germination of the pollen grain. Okay, children. Now what we see here is you are going to see the unequal division. Now, when the unequal division is taking place, so here what will happen is a bigger cell is going to be formed. One bigger cell and one smaller cell. So the cell that is the bigger one, it is going to be called as the vegetative cell. So children, यहाँ पे जो हमारे पास division है, you need to remember it is mitotic. तो यहाँ पे हमारे पास अनइक्वल माइटोटिक डिवीजन हुई है जिससे कि हमें दो अनइक्वल सेल्स मिलेंगे. लेट अस सी दोस टू अनइक्वल सेल्स. तो हमारे पास ये जो बड़ा वाला सेल है इसे हमने नाम दिया है वेजिटेटिव सेल. ठीक है, ये हो गया. अब वेजिटेटिव सेल के ही हमारे पास साइटोप्लाज्म में यहाँ पे एक हमारे पास छोटा सा स्मॉल अनइक्वल सेल है एंड दिस इज कॉल्ड एज द generative cell. Clear? Now, in case of the pollen grain of a 3-cell stage, what we are going to see is you are going to have the exine in the same manner. You will be having the intine. Then inside the intine layer, you are going to have the vegetative cell. But rather than having the generative cell, we are going to see the two male gametes. So that is the difference between the pollen grain of 2-cell stage and 3-cell stage. Now what we see children is a mature pollen grain at the time of shedding, that is here we talk about the dehiscence, is generally 2-cell stage. This is how it is going to look like. A surface view and enlarged view of the wall will show you that the exine is thick sculpted outermost layer, and a very important question from the boards and the NEET point of view is that the exine consists of or it is made up of a substance that is called as the sporopollenin. Then the intine layer is thin, smooth inner layer that is made up of pectin and cellulose. Children, one very important thing about this sporopollen is that since it is present in the outermost layer, that is exine, this sporopollen is going to be a highly resistant material. What do you understand by being highly resistant? Highly resistant material refers to whether there is any change in the temperature, pH, enzyme, any kind of electrical shock, this is not going to be affected by it. So as a result of which, this is going to be non-biodegradable. Yes. So they are not affected by the temperature, pH, enzyme action, mechanical action, any kind of acid or alkali. And since anything that is non-biodegradable, so it gives us the opportunity that you'll see that it undergoes fossilization. Now fossilization means when millions of years have passed, even in some cracks, crevices, snow, we can find the pollen grains in the intact state. So the vegetative cell, it is a large cell with abundant cytoplasm and a large nucleus. It controls the growth of the pollen tube. The generative cell are the small cells with dense cytoplasm and a nucleus. It divides mitotically to form two male gametes during the pollen tube growth. Next thing is children, the cell wall is called as the two layers, that is exine and intine, and for this we use the word is sporoderm. So here the outer thick and sculpted layer is made up of sporopollen that protects the pollen grains, whereas the inner layer intine is thin and smooth layer that is made up of pectose and cellulose. The germ pore are present in the exine and they are thin unsculpted areas through which the pollen tube emerges during the germination. So, now children, what you see is here is a wide variety of the pollen grains that I have brought to you in this slide. So number of pollen grains, can you see the difference? Each of the pollen grain is going to be different from the another. So wide variety of the pollen grains are from sunflower, morning glory, hollyhock, lily, primrose and castor bean. Next topic is pollen allergy. Allergy is some kind of exaggerated response that we see. So when we are talking about allergy and the pollen, there needs to be some connection between both of them. So what we are seeing over here is that pollens are the main causal agents that cause allergy in the body of the organism. One photograph that you see over here is children that of a weed. Weed is any unwanted plant that is growing other than the one that is cultivated. And since they are unwanted, they are competing for the space, for the nutrition, and for every other factor that has been required by the plants that are cultivated. So they needs to be removed, otherwise they are going to cause a lot of trouble. So the plant that you are seeing over here in this photograph is Parthenium hysterophorus. And since children, this is a scientific name that needs to be underlined separately. Okay. Now, why is Parthenium harmful? What we had seen is this Parthenium hysterophorus has been accidentally been introduced in India while we were importing the wheats. So means it is some kind of a contaminant that came in the country India with the wheat. Now it spreads rapidly and is now almost found everywhere. That is why it is ubiquitous, and as a result it keeps on growing and spreading rapidly because its natural predators are missing. And when the natural predators are missing, it has a full-fledged chance of rapidly spreading itself. Okay, now its pollen grains are highly allergenic and they cause allergies in different kind of individuals. So what is the effect of Parthenium pollen when pollen has been released into the air? Let us see. It causes pollen allergy, it triggers the asthma attacks, leads to skin irritation and respiratory problems, especially during the flowering season, and may be a cause of bronchitis, sneezing, nasal irritation, breathing, and difficulty. So, we need to be very much aware and we should be wearing the mask and avoiding. If you are having very susceptible kind of immunity, then we should be avoiding in places where is a lot of flowering is going on during the flowering season, or the best way to avoid is wearing of the mask. Now, what happens? How the pollen causes asthma or bronchitis? So, what happens is, children, the pollen in the air are present, and with the help of the wind, they get carried away and then they have been inhaled either through the nose or the mouth. And as a result, what happens is your immune system start to overreact. How does it overreact? Since these pollen allergens, they act as the foreign invading body, your immune cells, that is the mast cells or the basophil cells, they start secreting the histamine. And histamine is a natural chemical messenger that has been produced by the immune system to help your cells communicate and protect you from any kind of problem. So What happens when the histamine is being released either by the mast cells or the basophils? The airways become inflamed. Histamine causes the widening of the blood vessels, and it results in the more passage of the WBCs to the areas that are affected to fight the invaders. So as a result, swelling, mucus production, and the narrowing of the airways takes place. And finally, when you have the airways that have been inflamed and they are filled with mucus, there is going to be a lot of difficulty in breathing. The cells that have been present in the bronchial region or in the lungs, they are going to get inflamed and may become the cause of asthma or bronchitis. Even some people see certain kind of other symptoms like sneezing, runny nose, itchy eyes, etc. Then pollens जो हैं, they are abundant resource of rich amount of nutrition that यहाँ पे बहुत सारे बेटा तरीके के अमाइनो एसिड्स होते हैं, एंड अमाइनो एसिड्स आर द बिल्डिंग ब्लॉक्स ऑफ प्रोटीन. तो यहाँ से हमें अमाइनो एसिड्स मिल सकते हैं. विटामिन ए, बी कॉम्प्लेक्स, सी, डी एंड ई आर प्रेजेंट. बहुत सारे मिनरल्स लाइक कैल्शियम, मैग्नीशियम, आयरन, जिंक, पोटेशियम. बहुत सारे एंटीऑक्सीडेंट्स जो हमें बहुत सारे डैमेजेस से बचाते हैं. कुछ हमारे पास एंजाइम्स हैं, कोएंजाइम्स हैं, एंड अ लिटिल अमाउंट ऑफ carbohydrate, fatty acids, phytosterols, and flavonoids are present. So they are rich in nutrients and increasingly being used as food supplements in the form of the tablets, syrups, granules, anything. So they are going to be very freely available these days. Now, claim benefit is that they boost the energy and the stamina, they enhance the overall performance, stimulate the immune response, then improves the nutrition and vitality and helps in recovery and endurance. So because of that, you have a very good market of the pollen products. So we can say that it's a natural powerhouse of the nutrients. So when all these things, different type of nutrients and these leading to the increased energy, stamina and the performance, it has been used by both the athletes and the race horses people so as to provide them with the energy for doing these kind of activities. Moving on to the pollen viability, children, here what you see is this is your wheat, and here in this diagram you see the rice. So what happens is viability, when we are talking about, it is the duration from which or for what time of duration these pollen grains are going to be active. After the activity period, they are not going to be surviving. So here when we talk about the viability of the pollen grains, we need to understand that the viability depends upon two important factors. Number one is the temperature, and number two is going to be the humidity. These are very important factors in deciding the viability of the pollen grains. Now, what we see, children, is that in some cereals like rice and wheat that the photograph is also showing, here the pollen viability is hardly for 30 minutes. While in certain families, like here we talk about, this is a rose plant, and rose plant belongs to the family that is called as Rosaceae. So family like Rosaceae, then Solanaceae, in which you have the members like potato, tomato, even your eggplant, and the leguminous plants that is called as Leguminosae, or you call it as Fabaceae. So these kind of the families in which you see all the type of different kinds of beans, then rajma, chola, ये सब जो होता है, so these are all having the pollen viability that is going to be there for few months. So it is not going to be for a period of 3 months, it is going to be there for few months. So that is the difference between the examples that we have discussed. So this is all for today. Children, in this class today we have talked about the pollen, how the pollen grains are going to be produced, that is the process of microsporogenesis. Then we had talked about the pollen viability, pollen allergy. Thank you.